So far we have discussed the basal domain and the lateral domain and their specific modifications, and now we're going to move on to the apical domain and its modifications. Typically there are 3 modifications that are important for your course in medicine. These are going to be the microvilli, the stereocilia, and cilia, and we are going to discuss them individually. Right, so microvilli are apical projections that are made up of actin, and these are connected to the terminal web base at their base, and these absorb these perform the function of absorption of nutrients. All right, so for example, water or food that is being digested, for example, in your small intestines, these take in the water and food and absorb them in your intestinal epithelium, for example. Now, under the microscope, you would not be able to see the individual apical projections of the microvilli. What you would see instead is something like this. So something like this. See this border right here? This is called the brush border. Or this is also called the, brush. This is also called a striated border. And this is normally what you would see on a microscopic slide of the microvilli. So now a clinical correlate to the microvilli would be, a common example would be celiac disease. It's called a disease called celiac disease. Very common, especially for the USMLEs. Celiac disease is characterized by a sensitivity to gluten. So there's gluten sensitivity in your intestinal epithelium, which results in, for example, if you eat a gluten-rich diet, then it would result in the damage of this brush border here, damage to the microvilli, and it will lead to microvilli being damaged, and then it would obviously lead to malabsorptions because microvilli are being damaged and they cannot absorb food anymore, so there is malabsorption. And this malabsorption leads to many different kinds of pathologies. Alright, so next modification is stereocilia. Stereocilia, I'm going to use this photo right here. Stereocilia are again apical projections made up of actin. Just like microvilli, these are again made of actin, and these are also apical projections on the apical surface. These are usually present in the testes, especially in the epididymis, and let me just write that down. Didymis. And in the inner ear, where they act as well, the inner ear contains sensory hair cells, and these sensory cells contain stereocilia on their apical surfaces, which react to vibrations. So, for example, right here, and these would react to vibrations and change the flow of ions through the cell surface membrane of this hair cell, whatever. This is these are things that we're going to discuss later. So like all these functions of the stereocilia and in the testes, in the epididymis, in the inner hair, the hair cells, we're going to discuss them individually and how they work in the later chapters when we discuss specifically, for example, the male reproductive system or the auditory system. So for now, you just need to know that these are apical surface projections that are made up of actin, and they're present in testes and inner ear. Okay. And the next modification would be the cilia, which we have, I think, already done or looked at least once in the respiratory epithelium right here, and we discussed that this is the cilia, which is responsible for the motility of the mucus that is secreted by the respiratory epithelium. Now the cilia, let me just write that down, cilia, these are made up of axonemes. These are proteins called axonemes, and they perform the function of motility or motion. A very common clinical correlate, very important for the USMLEs and other licensing exams, is the Cartaginer syndrome. Let me just write that down. Cartaginer syndrome. This is also called the let's say, immaterial cilia syndrome. Because in this syndrome, the cilia don't work, for example, in your nose, in your throat, in your respiratory tract, and in your testes, which results in, for example, bronchitis and infertility and things like that. You will study that more in other subjects, but yeah, that's pretty much it for the apical modifications that we just did. The 3 modifications, where are they? The microvilli, the stereocilia, and cilia. Let's go head over to our neat notes and see what we have learned. Okay. So, we learned the microvilli are made up of actin, and they are connected to terminal web at base, perform absorption. Okay, keyword absorption. In microscope, they show up as striated and brush border. We looked at the image, and we discussed the pathology, the celiac disease, which is characterized by sensitivity to gluten. Stereocilia present in the testes and the sensory cells of inner ear. We will discuss their functions when we do those chapters individually. And cilia. And the last one that we discussed was cilia, for example, in the respiratory epithelium. And I forgot to mention that a special feature of this is that it is present on every cell of the body. So at least one would be present on virtually every cell of the body. There may be cells, for example, in the respiratory epithelium that have more cilia than just one, and these are made up of axonemes, and they perform the function of motion. And their mutation leads to the immaterial cilia syndrome, the Cartaginer syndrome. You may come across this in medgen or biochemistry. And yeah, let's move on. So far what we have learned is the individual characteristics of an epithelial cell. So let's move over here. So this would be an individual epithelial cell, and we've studied its lateral modifications, its basal modifications, its apical modifications, but we haven't looked at the epithelium as a tissue or a group of cells, and we're going to do that now. Now, as epithelium is a tissue, the epithelial tissue is composed of a group of cells, and these groups of cells, depending on their specific characteristics, differentiate as to which epithelial tissue they form. And these would generally fit under 2 descriptions, either simple or stratified. These are very simple words, which mean simple. This word would mean an epithelium that has a single layer of cells. So just a single layer that composes that particular epithelial tissue would be simple. So as you can see here in this layer, we have just one layer of cells, just one layer of cells, and just one layer of cells. This would be your simple epithelium. But a stratified layer would be any epithelial tissue that contains more than one cell thick, which is more than one cell thick, or it contains more than one cell layer on top of the other. And these layers are then further divided according to the shape of the cells that they contain. For example, squamous layer. Squamous comes from a word called squam, which means to press or compress. So these cells sort of look like they have been pressed from above, and these are very thin. And then in the cuboidal layer, you'd see cells that are cuboid-like. And then in the column layer, you'd see that this cell shape characterizes long columns. So a single layer of these columnar cells would be called a simple columnar epithelium. A single layer of cuboidal cells would be called a simple cuboidal epithelium. A single layer of squamous cells would be called simple squamous epithelium. And then more than one layer of squamous cells would be called stratified squamous epithelium. More than one layer of cuboidal cells would be called stratified cuboidal. And more than one layer of columnar epithelium would be called stratified columnar epithelium. Now, under microscopic slides, some epithelia may show characteristics of a stratified layer, but they may not be stratified. For example, in the respiratory epithelium, I will write the examples down in a short while. And these are called pseudostratified columnar. There's usually only one place where this this kind of epithelium is present, and this is called the respiratory epithelium or the respiratory tract. So inside the respiratory tract you'd find this sort of epithelium, and then transitional is also usually present in just one specific location, and it is the urinary tract. And transitional epithelium is an epithelium that sort of contains different cell types. So for example, you'd have a different layer of cells here, and then a different layer of cells here, and a different layer of cells here. For example, you might have cuboidal cells here, and then some columnar cells, and then you might have simple squamous cells over here, etc. In urinary tract, however, usually there are umbrella cells that are present outside. You don't need to remember this now, because we will do everything later when we do those specific chapters. So for example, when we do the respiratory epithelium or the respiration chapter, we will study this, and when we do the urinary system, then we will study this. So you don't need to remember all the details like umbrella cells or whatever at the moment. And I'm just going to give you quick examples of all these epithelium, just to give you a heads up of what's coming in the future chapters. So now there is a clinical correlate that is related with the respiratory tract, and it's called the metaplasia. Metaplasia is a term that is used for the change of one type of epithelium or cell type to another. So this is called metaplasia. And in the case of the serous-stratified columnar, in case of certain diseases like chronic bronchitis, for example, this pseudo-stratified columnar epithelium converts into more of a squamous nature, and this results in further pathology, because the cilial lining is being lost, and then it cannot further circulate the mucus that is being secreted, and it causes further problems in your respiratory tract. And this may arise from smoking as well. So this is why smoking is bad, people. And last but not least, we are going to discuss glands. Let me just write that down. Glands. All right. Glands are usually of two types: simple or branched. So simple would be just like this, a non-branched, and then you'd have branched. Like for example, here there's different branches that open into these ductules here, and then they are also characterized by their shape. So for example, they could either be tubular, like this one looks like a tube, but they could also be acinar or alveolar, which would be something like a sac-like shape. So a rounded sac-like secretory portion. And these glands undergo three different kinds of secretions. So the cells that are present in these glands would secrete the stuff that is being secreted by three methods. These are merocrine secretion, for example, holocrine, and apocrine. In merocrine secretion, you just have your normal exocytosis. If you're very familiar with that term, what happens is that inside, for example, a cell, this is the nucleus, and there's a lot of stuff. Anyways, some vesicles go ahead and merge with the cell surface membrane, and then the stuff is exocytosed out of the cell. Holocrine secretion would be something like the accumulation of stuff. So for example, this would be a small cell, and then it keeps on accumulating the stuff that it's supposed to secrete, and then it keeps on getting bigger and bigger and bigger, and at the end it just bursts. So cell lysis, and then it releases its contents, and this results in the bursting of the cell while releasing the contents. And then apocrine secretion would be something like, for example, we've got a cell, and this sort of pinches off a part of it, including its cytoplasm. So like it pinches just a little part of it, and this part then enters, for example, maybe this ductule right here, here, and is secreted. So these are the three different methods of secretion for the glandular cells. And now congratulations, we have finished our first chapter. Now let's head over to our neat notes and see what we have studied today. Starting off with the apical cell surface specializations, we did the Microvilli, Stereocilia, and Cilia, and then we did the types of epithelia and the examples of where these may be found, and we did the clinical correlate of the respiratory epithelium, the metaplasia, which is characterized by the conversion of one cell type to another, and then we did the glands and different secretion methods. Yeah, I forgot to mention that glands may be surrounded by cells called myoepithelial cells. Let me just go over here and draw like a myoepithelial cell over here. So I'm drawing like sort of a myoepithelial cell here. Let's assume that these blue dots are myoepithelial cells, and what they do is that they act as small muscle cells, and they contract. And then as they contract, they allow the glandular cells to excrete their secretions into the ductules. And a very common example would be, for example, in the breasts, in the mammary glands. We will study those as well when we do the female reproductive system, I believe. Alright, so that's pretty much it.